Hello, everybody. My name is Seungyeul Yoo, and I'm very glad that my previous talk has the similar motivation as I have. So we also believe there is many problems in sample labelings in the public domain data set. So my title is Sample Alignment, a Critical QC for the Integrative Analysis. So as we all know, we are now in the era of big data in biology. So we can generate and process a vast amount of biological data in a very quick time. And also we can profile different molecular features very easily, like genetics, epigenetics, and genomics and proteomics data. And we want to integrate this data to understand the molecular mechanisms underlying this biological system, maybe via the network analysis. So for this effort, several different consortium has been working to deposit large-scale data set like a cancer, GTEx, and also long genome consortium, and they put the data set in the public domain. And the problem I have, as our previous presentation, there is errors, and they can be sometimes critical for the integrative analysis. For example, let's assume that the gene 1, arbitrary gene, is epigenetically regulated by its DNA methylation. So it's obviously linear correlated with its DNA methylation. But if there is a pair of samples is swapped, then you lose your association. You might have a wrong scientific conclusion based on this observation. So to challenge this problem, I actually developed a systematic approach to detect these problems and correct them, so-called mode matcher. So in this paper, I used 3 different types of molecular data, genotype, gene expression, and the DNA methylation. Based on their cis association, I could manage to detect and correct this sample annotation problem. So I want to now explore in TCGA, which is the largest and most comprehensive cancer data set, and many of us actually are still using it for many of our different studies. And it's a good data set because it has a large number of samples, and it has a diverse types of molecular data. So if I can detect the errors in here, then as she mentioned, it's a really widespread problems. So how can we do that first? Actually, same as she mentioned, like we can check the gender very simply. So Y chromosome specific information can be used to infer the gender of samples from the gene expression or DNA methylation. For example, in this case, if I use a methyl proof information corresponding to Y chromosome gene, PRKY, this information can give us clear information of gender of samples in all 12 different types of cancers of TCGA. And you see the clear piles of 2 piles, female, which is background noise, and the males are colored in blue. And you can see the ovarian cancer and uterine cancer, which is a female control, and the prostate cancer is a male control. So it's very clear. If I use this information, we can actually detect the several profiles, platforms in TCGA dataset has the very simple problem of their gender provided by TCGA. So next one is, so then, okay, can we believe the barcode they provide us? So we matched the sample of different types of data based on those barcode information. So, so the underlying hypothesis is here. It's a different data types should be similar for the same individual than the others. Like here in type 1, for the sample 1 of type A, should be closer to sample 1 in type B rather than all other samples. So if you plot the distribution of those sample similarity, the sample with itself should be highly correlated. So make long story too short, also we can detect many of these barcode problems in 5 different types of, I just try 5 and I could detect almost all in 5. It's not really many. You see the 2% may not be really critical for the integrative analysis, but still it's a problem. And sometimes, as you can see, if you want a very subtype specific analysis, it can influence your data. Also, when I check the influence of that, when I remove those samples with problematic annotation and recalculate the cis strengths with the DNA methylation and gene expression, and you can see that the p-value has been changed pretty much. So also they can also give you different list of the genes if you want to use some cutoff. So the summary is there are errors even in TCGA. We believe it's very well controlled data set, and we can detect them Not many of them, but still we can do that. And so I believe the sample alignment is a critical step for this integrative analysis. So thank you for your attention. My poster will be there tomorrow at 8:46. We can discuss more if you're interested. Thank you so much.